Hello everyone. Today our video's topic is the recently popular technology, mitochondrial DNA sequencing. In this video, we will introduce what mitochondria are, what the mitochondrial genome is, and how this technology is carried out. So let's start today's video. Mitochondria are organelles present in most cells, enveloped by two membranes with a diameter of around 0.5 to 1.0 micrometers. The primary function of mitochondria is to supply the energy needed by the cell, often referred to as the cellular powerhouses. Approximately 80% of the energy required for cellular life activities is provided by mitochondria. Mitochondria possess their own DNA, mtDNA, and genetic system, but their genome size is limited, making them a semi-autonomous organelle. The length of human mitochondrial DNA is approximately 16,569 base pairs. In comparison to nuclear DNA, which typically consists of around 3 billion base pairs, mitochondrial DNA is considered compact. Unlike the Mendelian inheritance observed in the nuclear genome, mutations in mtDNA are passed exclusively from mother to offspring. If a mutation is present in the father, it will not be inherited by the descendants. This pattern is referred to as maternal inheritance because all the mtDNA in the fertilized egg comes from the mother's ovum, and any mtDNA from the father's sperm is typically eliminated before or after fertilization. Mitochondrial DNA possesses many unique characteristics and irreplaceable functions. Mitochondrial DNA sequencing is primarily conducted using traditional Sanger sequencing and second-generation sequencing methods based on DNA synthesis. Sanger sequencing relies on the termination reaction with dideoxynucleotide triphosphates, DDNTPs, and offers advantages in mitochondrial sequencing, including longer read lengths and high accuracy. Mitochondrial DNA sequencing involves the following steps. 1. Sample preparation. Derived from various bodily fluids, tissues, and cells, such as blood, oral swabs, skin tissues, etc. Typically, chemical dissolution and organic solvent separation steps are employed. 2. PCR amplification. Polymerase chain reaction, PCR, is used to amplify the mitochondrial genome, increasing the quantity of DNA sequences in specific regions. Typically, conserved regions of the mitochondrial genome are selected as target sequences for amplification. 3. Library preparation. The PCR amplification products undergo library preparation, involving steps such as end repair of DNA fragments, adapter ligation, addition of index primers, fragment size selection, and enrichment. 4. Sequencing. The Illumina sequencing platform can be employed for bridge amplification and stepwise addition of nucleotides. In this process, the single-stranded DNA undergoes multiple cycles of reactions where terminator molecules bind to the target DNA and nucleotides are added one at a time. Fluorescent signals are then recorded by a highly sensitive camera, converting the obtained information into sequence data. 5. Data analysis. Initially, low-quality sequences are filtered out through data quality control. Subsequently, sequence alignment is performed by comparing the short fragments obtained from sequencing with a reference mitochondrial sequence to obtain reliable sequencing results. Finally, the comparative results are analyzed to identify mutation sites, construct mitochondrial phylogenetic trees, analyze human origins, etc. Statistical methods are employed to validate and interpret the results. The advantages of mitochondrial sequencing technology lie in its high copy number and haploid nature, providing abundant genetic information through the accumulation of mutations and replication in numerous mitochondria. Additionally, mitochondria exhibit multiple codons and interactions with the nuclear genome. Variations in the mitochondrial genome are associated with the occurrence and development of certain diseases, such as cancer, neurodegenerative diseases, cardiovascular diseases, and others. Mitochondrial genome sequencing has applications across various fields, including the diagnosis and research of mitochondrial and neurological diseases, studies in human evolution and anthropology, forensic identification of individuals in forensic medicine, and the development of drug and treatment strategies. Mitochondrial genome sequencing plays a crucial role in areas ranging from disease diagnosis to evolutionary biology and environmental research.